Drosten really needs no introduction, I think, to any of you who work on coronaviruses. He's been leading, conducting world-leading research for many years now in the field of coronaviruses, as well as other families of viruses. And it's an absolute delight that we've got Christian here today. And I think I need to go on talking for another minute while he gets his microphone sorted out. But yeah, I'll hand straight over to Christian, who I'm sure you're all here to listen to rather than me. Thank you.
Yeah, thank you. Thank you, James, for the very nice introduction. And good morning. Yeah, so we were stuck a bit over the pandemic, so we couldn't do a lot on MERS, even though we started before. And this is really an interesting virus and interesting epidemiological enzootic situation. And we are trying to catch up a little bit now after I mainly have been pulled in this ugly intersect politics and media. So I'm still not fully out of that, but let's see how we go. So we will discuss a little bit the evolution of MERS as it goes on in Africa and the Arabian Peninsula. So most of you will know, but for those who don't, so this is a coronavirus, a beta coronavirus, MERS-CoV subgenus, that is widespread in dromedary camels, where you have dromedary camels, except a few places like Australia or some parts of the Canaries. Else, where you have dromedary camels, you have very high antibody positivity in the animals, especially, of course, in the adult animals, and it's very widespread as a virus in the young and the calves. It's acquired in a zoonotic manner, mainly in hospitals where it becomes visible, but it also happens in the community. So about 0.1% of the population in Saudi Arabia, for instance, are seropositive. If you look at adult people there, which amounts to maybe 10,000 infections per year that are not reported. And there are also a few recorded human cases in Africa, but the number is really small, and it's still a little bit enigmatic whether this really happens and whether this really has a relevant dimension. But in animals, it's much clearer. So we have this low density camel keeping towards actually into Uganda. And then as you move north into the more dry areas in Eastern Africa, you have this nomadic keepings that become denser and denser as you move into Somalia and the Horn of Africa. And from there is intense camel trade. And then in the Arabian Peninsula alone, around the holy places in those 2 weeks of the Hajj pilgrimage, about 40,000 camels are slaughtered as a religious sacrifice animal per year, only in these 2 weeks. And then it goes on around the year, and there's very intense camel farming. And then camels are also kept as leisure animals, a little bit in the manner as like we keep horses for leisure purposes, they keep camels, but here it's the women in our area, there is the men, right? So if when you're a man of middle age and you've made it to some economic wealth, then you start thinking about keeping camels, maybe with a friend or two to keep 1 or 2 camels somewhere. This is quite widespread, and it's a very high cultural value. And this leads to unnoticed spillover infections to mainly young male people in the Arabian Peninsula, and to noticed infections to older, also mainly men, who turn up in the hospital and there cause nosocomial outbreaks. So if you and in the households, the situation is much less well recognized. So there is household transmission, but it is less effective. This is not a perpetually transmittable virus, but it's more like an occasional detection when you go and look for it. And this is also somehow reflected in the reporting. This here, this big peak, is the Jeddah city outbreak. So this was the most notified outbreak, actually. This happened very short before the big Ebola crisis in West Africa, and drew a lot of attention by international organizations like WHO, actually to Jeddah, when the outbreak in West Africa was taken off, and it caused some delays there in terms of international public health attention. But this really deserved attention because this was a nosocomial outbreak across a big city in Saudi Arabia in many hospitals, and a situation where you could summarize like it is quite normal that 3 to 4 generations of human-to-human transmission occur. So this is, of course, a situation where a virus can be selected for adaptation to humans when it is a normal situation that you get a few generations of transmission in humans. And the same thing also happened around 2015, where a new virus lineage came in, and this was then transferred to South Korea. In South Korea, we had the same observation, several generations of human-to-human transmission in a hospital system. So that actually is something we should really look at when we are concerned about a virus that might adapt to human transmission. So let's look at the virus in the Arabian Peninsula, mainly with a focus on the more current virus. And this starts in 2014-15, where a new lineage termed the lineage 5, or then the novel recombinant clade, took over. So this is GenBank entry, so we're not in the range of 14 to 15 million like in SARS-2, but a few hundred in the databases. But still, you see how the old viruses disappear and the new virus comes up. And we were lucky to be able to work with a colleague in Jeddah, with Dr. Issam Azhar, who is still interested in camels and MERS, and he's sampling with his team. And we're collaborating with him to characterize what's going on now after the pandemic, because there was quite a notification and activity gap over the pandemic, like in every other country. Much of the research went into SARS-2. So after the pandemic now, we have a very colorful picture. So these are phylogenetic clades. You see how they are distributed. This is a pan-mictic situation, we could say. So a situation where the virus is distributed all over the country. Where you look at commercially kept animals, you will find several virus lineages present. It's all a mixed picture. And there is high rates of positivity. Look at these numbers, right, in different sampling sites. This is a tree. This is the beginning of the B5, the novel clade. So these are the parental clades here. And only here in the top of the tree, these are the post-pandemic viruses that we are now looking at. So this is just from here is one diversity growing like a ladder-like clade. And from here, then new diversification with a lot of internal recombination, actually. So these lineages are now becoming more and more differentiated, it seems. And interestingly, more like a curiosity. So this type of recombination doesn't seem to wipe away the time signal from the tree, like sometimes we observe in other trees. So there is not much evolution going on here. And we still have a quite perfect time signal, even looking at all the sequences in the tree, including the recombination events. Still, we have a very good time signal. But then here with the lineage 5 sequences, this is really a perfect time signal still. So this recombination acts on the tip of the tree, and it's really recent. But it's quite prevalent. You can demonstrate how these lineages cross over. So this is all within the lineage 5. And of course, even if we can't derive any phylogenetic novelties here, one notion is quite clear. For such prevalent recombination, you need quite a number of infections, right? You need many animals double infected. And this matches the high detection frequency that Issam had in his animals. So a small look at the phenotypes. We were active in Saudi Arabia at a time when just the genotype switch occurred. So before and then in 2014 into 2015, we received samples mainly from humans, from hospitals, and did virus isolation trials, and then picked some 40 isolates that were representative of phylogenetic clades. And these also included the lineage 5. So this is the lineage 5, the recombinant one, and these are the two parental lineages, lineage 3 and 4. And for the data that I'm going to show, we are using these viruses, so all of the red ones, and we pick isolates from these clades and replicate the experiments so that we have a very good insight into the existing diversity of the viruses, like how they behave in the lab. And if you put them in cell cultures like Vero cells, there is no difference in growth. In callus cells, we know there is some interferon activity. We, at this point, already was suspected that this is linked to cytokines. So the lineage 5 shown in red grows better in callus cells, also in caco cells, polarized callus cells, better growth. We didn't consider temperatures at this time. This was really, we looked for cytokine-related differences. Also in more sophisticated models like human airway epithelium, human lung explants from peritumor areas from cancer patients, or from camel airway epithelium, you see how the virus grows better. Yeah. So in all of the instances, there seems to be more replicative fitness for this virus, for the lineage 5, also more spread here in the terminal bronchial area in such tissue models of human lungs. And just to make sure it's not related to entry, we did some entry-related experiments. Stefan Püllmann did this for us. So there is no difference in VSV-based entry assays, as you can see here. So the red one always is the lineage 5, and then there's the two parental ones and the EMC, the reference virus. All of them enter to the same degree, including in the cells we tested. So our phenotype wasn't based on entry. This is DPP4 binding assays per fax. And also here, it doesn't seem there are any relevant differences in DPP4 binding. Also using full virus, like synchronized infection, cold cells, virus on top of the cells, then out of the fridge in the incubator, 73 degrees, and then measuring the onset of N gene transcription after 4 hours as opposed to the control. So this is a genome control, and this is the N gene, and this is then de novo synthesized N gene transcript after synchronized entry. It's all the same for these different virus lineages, right? And if we inhibit this by entry inhibitors, this goes away. So it seems no relevant entry phenotype. Whereas the cytokine induction seems to be different. So this is after the same seeding dose, the resulting replication in the experiment, and then with this higher replication dose, still a higher induction of these messages, including some directly induced ISGs and then cytokine genes, and a higher resilience. So if we same observation here, constant seeding dose here for the viruses, but then much higher replication, and then also more robustness against interferon application. So this is 25 units. This virus is quite sensitive, like most coronaviruses. If we use ruxolitinib to inactivate the JAK-STAT signaling pathway, this levels off, right? So this is these cells, the virus here, with a higher fitness, and then this fitness difference lost when using a JAK-STAT inhibitor, and well, not as clear a phenotype in cells with a receptor knockout, but still the trend. Okay, now these are the founding lineage 5 viruses that were isolated, let's say around 2015, so when lineage 5 was just coming up. We are now after the pandemic. So these are now the post-pandemic viruses, and we are looking at 2 exemplars here. Actually, we're currently working on a completely genetically representative sample of rescued viruses, but we already have some preliminary insight here. So these 2, the gray and the orange, are 2 exemplars. And you see how they differ now, right? So these viruses differentiate a little bit. So these are these are two different cell lines, and you see this is well, a virus that we'll discuss later, one from Africa. But this is the parental, the original one, the old lineage 5 virus. And here are two recent ones, and one of them has a lower fitness. And a similar observation was just published as a preprint by Malik's group, Malik Paris from Hong Kong. He sees the same thing, right? So they also work on different, more recent lineage 5 viruses, and they are not all the same in their tests that they do. So really interesting preprint that I can recommend. It just came out 2 weeks ago. And yeah, so there is no obvious change of replication in post-pandemic MERS lineage 5 based on these virus constructs that we make. So this is the EMC, the control virus, and this is the enhanced replication of the old lineage 5 virus, and the new lineage 5 virus here replicates at the same degree. So here there is no change. But some of the more recent B5 strains are more sensitive to interferon treatment in callus cells. So if you compare, this one is the old one, this is the new one. Both of them are lineage 5 virus. This is the comparison virus, the positive control virus, EMC. So really a phenotypic differentiation, quite a big replicative difference, more than 2 logs under a given interferon concentration, while they are the same without interferon. Very interesting phenotype. So this virus is really moving. So to summarize, this lineage 5 associated phenotype up to now seems to be associated with evasion of the interferon response in some sense. And we are now looking to understand better how this works mechanistically. I'll skip some of these data for the sake of time. And would like to go to Africa, to the enzootic virus there. Also here we have some more recent insight. One piece of data is based on a study we conduct, and this is the group of Marcel Müller in the lab. He with his group started a big fieldwork activity in Kenya, and what you see is sampling over 1 year in time, and what you also see is 2 periods of virus detection: September, October, and then January and February. These are animals sampled as at a big slaughterhouse, basically, and this is the caption, caption area of this slaughterhouse, and it's a considerable fraction of animals who are RNA positive. These are adult animals slaughtered, still RNA positive. Actually tells us something that there should be some epidemic activity, and it seems that this is somehow related to drought periods. So we have in September, October, and in January, February, and March, the dry seasons. And what we think is these are not feral animals. This is nomadic camel keeping, but the animals have a very large range that they can roam, and they come to drink to water holes. And of course, the drier the conditions, the lesser the water holes, the opportunities for them. So there is more encounter between the animals. And the reason here there is no infection in this long dry period is that the camel calves are already through the worst in this period. So they are born around winter, and there is an infection peak here, and probably the newborn camel calves are immune by this time. So this is what we, our hypothesis that we are still trying, of course, to fill with data, but it's based on prior observations we made long before the pandemic, looking at camels in Dubai, where the pattern is very similar. Okay, the other question is human infections in Africa. Is there really no human MERS in Africa? Well, there is this old study we did in 2016 already, where you can say, well, if you test by ELISA, and if you go to villages, and if you go for people who have contact with all sorts of animals, including camels, then there are some ELISA positives. So the blue indication means they also have some signal in immunofluorescence, and then there are 2 red ones, they are neutralization positive. So it seems there is human infection, but it's quite rare. If you do some statistics, however, you see that, well, this is about 2 neutralization confirmed in 1,000, and this is not much less than in Saudi Arabia. The frequency, if you look at normal human population, is also not much higher in Saudi Arabia. And this is more recent serological or immunological work, and it's the same observation again. If you look at camel-exposed abattoir workers, you see there is an ELISA response in quite a few of those, and then one of them is confirmed by neutralization test. So it's not a frequent thing, but it's also difficult to assemble very large cohorts here. So imagine this to be not 48, but 4,000, then you would already be quite impressed because there is quite a number of infections, right? So we are often fall to these biases. If you go and do field work, seroprevalence studies in the field in villages again, you are sometimes surprised by the seroprevalence you find, like in the full percentages, and then even here, but this is probably an artifact. We don't believe this, like a quarter of people seropositive. Something must go on here. Many of those can't be confirmed by neutralization test, even though they have very clear ELISA reactivity, and this is not cross-reactive with SARS, right? Of course, this is all post-pandemic. Everybody has been infected multiple times, but there is no correlation at all with SARS titers. This is RBD-based ELISAs, and it's really quite specific. So we don't understand what's going on here. Many of those people don't have neutralization titers, but ELISA reactivity is prevalent. Cellular immunity is another, let's say, success of the pandemic, right? So with the introduction of those simple format cellular immunity tests like interferon gamma release assay formats over the pandemic, it's now possible to do cellular immunity testing close to the field, and we've adapted one of these tests to MERS. So trying 2 different peptide pools here, and then this is a plain, well, fresh blood stimulation. You take the full PBMCs, stimulated in fresh blood, and then with a mitogen control, you test for interferon gamma release, and this you assay by an ELISA test. That's all. It's just stimulating activated cells in peripheral blood. Probably this will go away after 2-3 years post-exposure, but people are re-exposed, so there is always some circulation of activated cells. And then so these are SARS coronavirus controls. There is, especially in this pool, there is no cross-reactivity. And then if you use people from a phase 1B vaccine trial, MVA MERS vaccine trial that's going on in Hamburg, you see how nicely they react with this test. And then if you apply this to people in the field, 3 screens, this is SARS controls here on the right-hand panels, and then the left-hand panels is in the 3 sampling time points, the MERS reactivities. There are always some, right? These are slaughterhouse workers. There are always a few that react. So this is a real thing. There is MERS infection, but it's a bit difficult to define serologically, and we've never been able to catch somebody who is virus positive currently. But this is, well, field conditions. It's not easy to have these numbers that you need. Also, what we see and what others also see is there is not a very good correlation between the cellular immunity or cellular, let's say, stimulation and the ELISA reactivities. So this is the same people, very bad correlation actually. So there are people who react, who have good cellular reactivity, others have good ELISA titers, not a good correlation. That's the situation. Okay, a little bit on genomes and function of these viruses. So we started working on this based on camels imported into Jeddah Harbor. A good opportunity to sample from African viruses, because these are all African viruses coming in there. And look at the positivity, right? So this is 13% of them positive. All of them are clade C viruses, so they're African viruses. And then local control camels, 50% of them positive, all Arabian virus, all clade B. And we can summarize the situation such as African genotypes, they seem to reach the Arabian Peninsula steadily, but they are never detected in local camels. So African viruses don't make it in KSA. The local viruses seem to compete them out. It's a bit unclear. We can't mechanistically explain, but this is the observation. Nobody has ever sequenced really an African virus from an Arabian camel. There is one little exception, but this might be an odd one out. One sequence that was detected in one of the Emirates, probably based on a direct importation. Okay, several of these African viruses, if we try to ascribe properties to them, have deletions, especially the clade C1 virus. They have many deletions in the accessories, and most of them in open reading frame 4b. There are different patterns of deletions, many affecting the nuclear localization signals, already the entire gene, and then others affect the phosphodiesterase domain. So we modeled this a little bit by making mutations in an infectious clone, knocking out the nuclear localization signal or the phosphodiesterase. And I will show data only for this one because the phenotype here is minor for the phosphodiesterase. So if we knock this out, best visible here where both of the domains are knocked out, and compare this against the control virus, we see how the protein 4b goes to the nucleus to basically block a nuclear pore that prevents then the importation of cytokine transcription factors. And where this doesn't happen when we knock out the nuclear localization signal, the fluorescence is completely distributed in the cytosol. Yeah, and then growth studies show the expected phenotype. So there is attenuation when we knock out these nuclear localization signals. Here in Kalu, only visible at 16 hours, but in H1299, another human lung cancer cell line, the phenotype is more pronounced. And I have to say the virus grows very slow in these cells, so it gives the phenotype more time to develop, while here there's already a lot of cytopathogenicity. Yeah, and mechanistically it works. So this is NF-kappa B staining or IRF3 staining, and you see with the intact nuclear localization signal, the protein going to the nucleus, oops, sorry, and the NF-kappa B transcription factor staying out, and then the reverse pattern once you eliminate the nuclear localization signals, then the virus protein stays in the cytosol and the cytokine is induced. So the transcription factor goes to the nucleus upon stimulus. And for IRF3, it's the same pattern. So we don't have to go very much into the details here. But also here you see how with the deleted nuclear localization signal, IRF3 goes into the nucleus to then, of course, induce type 1 interferon. And as you can see in terms of mRNA expression, how the cytokine genes go up more with the deleted virus. Yeah, so it's not a very big, impressive phenotype. For some of them, yes. For some of them, no. But this might, and as I already said, there is not such an obvious phenotype with the deletion of the phosphodiesterase domain, which acts via the OAS RNase L system. But there seems to be some hints that the deletion that these viruses acquire there in Africa might attenuate them. And that might be one of the explanations why we don't see these viruses in Arabian camels. So we were speaking about these viruses here. So these are the African viruses. And we have some more recent ones now sampled in Kenya. They are here, clade C22. This is C1. And these are somewhat different. These are the more recent exemplars we have. So if we put them in human bronchial epithelia, these are two controls. This is the EMC and this is the lineage 5 from Saudi Arabia that we already discussed. Grow them in bronchial epithelia, we see there is much less growth for these Kenyan viruses. These are two exemplars of the more recent Kenyan viruses. This is also reflected in plaque phenotype, plaque size. And also in human nasal epithelia, it's a very big difference, right? So it seems they start to replicate a little, but then this is the Arabian virus, and here is where the African virus already loses the air and doesn't continue to really replicate. Yeah, this seems to be linked a little bit to temperance expression. So this is vero cells. This is also vero cells. another, well, subclone, but with a tempress transgene. And here you see with the tempress transgene, the difference is much less than without the transgene. So these are the cells that we used to use in the lab before the pandemic. Now everybody's switching to these cells after the big observation of tempress usage and SARS-2 with the furin cleavage site. MERS, by the way, also has a furin cleavage site, but it's not a polybasic one. It has just 2 basic residues. But it seems to use tempress, seems to be important for MERS. And yeah, we are looking at some features. So we are scanning these mutations currently in the spike, and there are some preliminary data. For instance, this is an interesting mutation in the RBD motif that also showed up during the Korean outbreak with another variant. And this is a mutation in a catepsin cleavage recognition region that might also affect entry. When we so this is the D510Y, and this really seems to, or is predicted to, affect the charge potential around the receptor binding moiety. So there is a negative charge potential with the wild type, and then with this one observed in all of these novel C22 viruses from Kenya, there is a lesser negative charge potential around the receptor binding pocket here. And well, whether this is based on immune pressure, we don't know, but we can look at the phenotypic consequences. So this is an entry assay where we have on Y the ratio between viro and viro-tempress. So it's in fact the tempress dependence that we see, right? We see how much better the virus enters when it has cells that have tempress as a transgene. Now, what's the time? Sorry. Yeah. And so this is the ratio 1, and then you see how this virus is more dependent on tempress. This is based in pseudotypes. And then for the other mutation, this has no consequences on tempress as expected. So we really see more tempress dependence of this virus. And then when we do inhibition experiments, it's the same observation. So this is the control virus. This is one of those Kenyan viruses. And you see how with increasing concentration of camostat, a tempress inhibitor, the replication goes very low for the Kenyan virus, and there is a residual replication, residual infectivity for the EMC virus that can be, let's say, sacrificed when applying additional E64D, so a catepsin inhibitor. So it seems that the Arabian viruses, they slightly use also the endosomal entry. You can separately inhibit both of the entry patterns, but you can't for this one. For the Kenyan viruses, this entirely depends on tempress. Okay, then some fitness effects of these. So these are now live full viruses, recombinant. And what we are looking at is spikes of 2 different of these more recent Kenyan virus in the backbone of the EMC control virus. And what we see is that the spike by itself reduces the fitness. So the Kenyan spike is less fit. Everything else here is identical except the spike. These are full chimeric viruses. And there is also a backbone dependent effect. So if we do the reverse constructs, if we take a full genome of two of these Kenyan viruses as infectious clones and give them the spike of the EMC virus, then it's the same, then it's a very much different situation here. So this is the same spike with different backbones, and this is the backbone of the Kenyan virus, where we have a significantly reduced replication and also increased sensitivity to interferon. Okay, so to summarize, I'm through with what I wanted to show you. Here, for the more recent viruses in the Arabian Peninsula, we have a growing phenotype diversity. It seems they are not all the same anymore. The selection here and the differentiation takes place in camels, and the properties that these viruses show in humans are differential. And the early lineage B, the fitness increase that is observed here is probably based on cytokine effects rather than spike-based effects. For the C1 African viruses, the ones we looked at first, there is an attenuation based on the loss of the protein 4B, which is again based on, again related to interferon effects, interferon evasion. And for the more recently studied viruses from Kenya, well, we have indications of human infection from the field, but there seem to be quite some differences, particularly linked to entry. So this virus seems to be more temperance dependent. So it's really the question whether these viruses are fit enough and virulent enough to really infect humans and cause transmission chains. So this remains to be determined further. Okay, so the contributions here, this is the Mwangi and Rito labs from Nairobi, 2 different labs, an immunologist and a veterinarian epidemiologist. Then Marcel from my lab, my own group, and then Esam Azar from King Abdulaziz in Jeddah. Thank you.
So much. This was like an incredible presentation. We have time for some questions. Yeah, Federico.
Well, thank you for the fantastic talk. So you said that potentially the difference between clade B and all the others is in cytokine effects. Have you pinpointed certain candidate mutations in that clade that might lead to a decrease in cytokine effects, or think that?
Yeah, well, we're currently working on this, obviously. So we are shuffling the genomes. We see indications mainly for the NSP3 region where we have stronger phenotypic effects. Of course, there are also some phenotypic differences associated with the accessories. We're taking out these accessories, exchanging them. Yes, there are differences, but these differences are often minor.
So maybe it would be like a cumulative effect type of.
Yeah, it's certainly cumulative. Yeah.
Here. Yeah, very nice talk. You saw that the lineage 5 have increased the fitness in humans and suppressing the cytokine response. Have you seen that in camel cells as well? I'm just wondering how the current lineage 5 evolving in camels, whether they also evading the camel response.
Well, we see similar phenotypic presentations in growth, but we have not really done, let's say, systematic studies of the interferon response in camel cells. Most of the cell lines available are really not close to primary cells, right? Several of them look like fibroblasts, and we don't really feel comfortable working with these models. And so currently, I mean, it's really an adventure, right? We're currently collaborating with the zoo in Berlin to get nasal brushes, and yes, they grow sometimes. Yeah. So that's the state of the affair there.
The camel cell line.
Yeah.
So Miles and Paul.
Just trying to make sense of the sero-epi data from Africa, and we talked a little bit about it yesterday.
Yeah, I thought about your remark yesterday. Yeah.
So you see these relatively low numbers of hits in the high-risk populations, i.e. the camel herders and the abattoir workers. But how does that compare to the general population? I mean, is it just the general thing you're seeing, or is it risk associated?
Yeah, I mean, so the seroprevalence data that we saw in these camel workers were really like in the percent area, like 2 or 3%. this old study in Kenya that we did, this was more like going to villages and sampling people who keep some animals, yeah. And they were in the range of 0.1, 0.5%. So it's clearly lower, but all of these numbers don't really make for valid statistical comparisons.
Yeah. I mean, I've said before, in our work in West Africa, where there are any camels. I mean, we do see this level of a significant level of serology positives, but it's not neutralization positive.
Yeah.
And so we don't think that it's MERS, but it's something else similar to MERS that hasn't been identified yet.
Yeah, that would be very interesting. I mean, I would be happy to look at things like this, and maybe we should also look in our samples. Yeah, but I mean, there are other explanations, especially with affinity-based serological assays. You could see cross-reactivity based on technical things about the antigen and so on. We are trying to clarify this.
Miles has sort of made the point I was going to ask. If the discrepancies, whether it was saying there's another MERS-CoV virus knocking around, or is it just noise?
I don't know. I mean, of course, we now learn more about MERS-CoV viruses. There are all these HKU4 and 5 related viruses, some using DPV4, some using ACE2. Who knows? Yeah. We've never come across something like this. So, I mean, in Africa, we've really focused on sequencing camels, but a lot of this is based on hypothesis-free sequencing. We found like the bovine coronavirus and these 229E related viruses at quite some frequency, but no other coronaviruses yet.
There's some question here on the right, no? Yes.
Great talk, thank you. I know you said you've not seen the African versions of MERS in the Arabian Peninsula, but have you ever seen the Arabian versions in Kenya? And if not, why not? Is there no transfer of camels that way, or?
Yeah, well, there is much less trade. I wouldn't say there is no trade at all, because camel breeding is also a practice, and there is a science behind it. And of course, the Arabian camels are, let's say, thoroughbred, right? And they are interested in these camels also in Africa. So I wouldn't say they are not importing camels from the Arabian Peninsula into Africa, but the big trade goes into the opposite direction. But so no group has ever detected lineage B viruses in African camels.
Interesting, thank you.
Thank you, James. Great talk, Krishna. Can you comment on host range for MERS virus? Because I think in SARS, we saw it spreading into other animals as well. Have you seen it spread in other animals?
No, there is no convincing data for any other, especially in livestock. There is no other species that is really infected with MERS. And also, I'm not aware of any wildlife animal species that is reasonably infected in terms of, let's say, field-based seroprevalence. Also, no convincing lab data. So you can cross-infect other camelids. This virus seems to be quite adapted to the camel.
One question on the right.
When we got one at the back, and then we should.
Thank you. Over here. I was just wondering if you could speculate on what you think the selection pressures might be that allow loss of function of a gene like ORF4B in the African viruses. So what could be going on in the ecology of the camels or the bats that allow the loss of function of a gene like that?
Well, probably it's simple. It's neutral evolution. So it's bottleneck effects in Africa. So if on the African continent, you have this area of very dense camel keeping, and then it thins out a lot into West Africa. And this is really where you find those viruses with the deleted open reading frames. They are typical, for instance, in northern Nigeria and other areas of the Sahel, where there is camel trade, but much less dense. So a lesser degree of lineage competition in the viruses, and we see the survivors of loss of function mutations that survive anyway, and that would be competed out by competing lineages without the loss of function in a more dense situation. That would be my simplistic explanation.
The gentleman at the back.
Yeah, thank you so much for the presentation. Myself, Ahmed Al-Barak from Saudi Arabia. So I just trying to maybe comments rather than questions. So for MERS, as Christian mentioned, there is a notification gap happened to 2018. And the 3 points I need just to share with the community here. First is the testing of MERS in Saudi Arabia increased, and we integrate MERS within the surveillance that we do in the respiratory viruses. And there is no single case detected among the population. So there's no circulating infections in the community. And we are sure of that because we are testing, last year we tested more than 25,000 community participants in the primary healthcare centers, and none of them turns to be positive by PCR. And also we testing the population of the hospital, very severe cases in the hospitals, and usually there is no case except few cases that has been diagnosed and reported. So I can say it's a fact that MERS is not really, this is a real drop in detection of cases and notifications. Going back to the serology, again, I don't think zero prevalence is a great tool to assess the transmission of MERS within the community in terms of false positivity and cross-reactivity with ELISA and other things. So we did a recent survey. Again, we found positive serology, and then we do the SNT and neutralization. It comes to be negative. So this might be another explanation for that. For the genomics part, we did also some genomics from the positive human cases, which is missing from the database. We will submit very soon. And we compare that with the camel cases and the recent ones. What we found out, the virus in the isolated from the human cases is not exactly the same as the virus that we isolate from the camels. So our initial interpretation is really maybe the virus is more adapting now to the camels more, and definitely will be more fit in camels and maybe in the cell lines, but maybe less fit in terms of transmission to humans. Because most of the viruses that we characterize and sequence from the human cases, they are not exactly similar or relatively similar to those we isolate from the camel cases, except in one case where the patient acquired the virus from his own camels within the farm. So still, there's a lot of questions that we need to answer. Really enjoyed presentation by Christian, but I think there is lots of questions that need to be also looked at and answered. Thank you so much.
Christian, do you want to conclude with any comments? Because I think we should wrap the discussion up.
Yeah, well, probably the most also related to your comments, the most interesting and relevant question, I believe this is not finally answered, is whether the SARS-2 pandemic introduced any type of population background immunity now, right? So it's a different subgenus. It's not immediately visible in those coarse type cellular immunity tests that we did, but there might still be some cross-reactivity if you look closely. And that might be another explanation, together with this alternative explanation of the viruses. Well, you say adapting more to the camel, but I would say they become more diverse, and there are lineages that are not as fit in humans, it seems, while they may be as fit in camels as before, or even better. So this is possible, but it's also possible that there is some yet unnoticed population background immunity introduced by the pandemic, which would be a very good message. We would have to look close for this.
What a fantastic discussion. Thank you all very much for the questions. And particularly thanks to Christian, who's come here to deliver this keynote. And we're really, as I said before, we're really privileged to have him here. And what a great talk. And Christian, thank you very much indeed.